Well, thank you very much, Shiva, for the very introduction. It's a pleasure to be here today. I've been in India now, but this is the first time in my life that I come to India, and it's been quite an experience. Started with a conference organized last week, basically, the International Biogeography Society conference, for which I gave a small talk and met some very interesting people. Got to learn a lot about the research being done in India, which is great because, as you probably know, there's quite of a big divide between what happens in the Western world and the Eastern world, and it even reaches us, I think, scientifically speaking. Sometimes we don't necessarily publish the same journals, and anyway, so it was super interesting. And then I went on 5 days of expedition with Shiva and his friends and colleagues. Did some safari, went to see the Shola Mountains, or the Sholas in the mountains. Learned a lot about these ecosystems and how biodiversity is maintained in these ecosystems, the main conservation issues, poaching, all these things which are not as big of a deal in North America, I would say. Then we went and visited the freshwater swamps where Shiva works, did his PhD work, and that was quite amazing also, and just makes me want to come back here and do more research. So it's been great. I want to thank Shiva the main reason I'm here is because Shiva insisted and was very enthusiastic about me coming and made it very welcoming. It's been great. So it's so much fun. And now I'm excited about telling you a bit about some of my research. I do a lot of things. I'm a community ecologist and a biogeographer. So I do some of the things I do at a very small scale, trying to understand how species will coexist, even in a small forest patch. And then another part of what I do is more about trying to explain large-scale pattern of biodiversity. And so why is biodiversity unevenly distributed on our planet? And so my talks is a bit about these two things. And I actually started my career in science as an entomologist and especially an ant ecologist. That's really what got me going. I was an undergraduate student at McGill University in Montreal, Canada, and just one summer started paying attention to ants and thought they were the most amazing thing in the world. Then I went on and spent 15 years working on ants after that, and I did an entire PhD on that. And although my postdoctoral studies were on things like building the Wallace map, I kept working and collaborating with ant people. And I will probably never stop. I have 3 graduate students working on ants now on various questions, but always relating to coexistence and biodiversity. So my talk today is pretty much kind of a summary of the last 10, 15 years of work that I've done on them, trying to understand how ants coexist together, but also trying to understand how biodiversity varies through space. And it turns out that coexistence and how biodiversity is distributed in space are related to one another in many ways, right? So you kind of need to understand coexistence to really understand what maintains biodiversity and what creates shift in biodiversity. Okay. So the main question that we ask in my lab, or that I have been asking for all these years, is what determines the structure of a community? And so the community here, is described as white dot on a global map, right, the map of the world. And the main question that we're asking, who is in there, how many, and why? And it seems very simplistic, but that's really the bottom line of what we're asking. And more specifically, what we're interested in here are the processes that are shaping the structure of local communities. So, in other words, what are the processes that determine how many species you have in there and what the composition of species is in there, right? How many is composition, Y is about how many. It's gonna take me a few minutes to get the whole of this pointer thing. Okay, I need to point down. All right, so I just explained that. Over the years, I realized that to really get an understanding of who's there and how many, you need to think about processes happening across different spatial and temporal scale. And so I started my PhD by looking at very fine spatial scale processes, how ants coexist in local patches, and then many years later ended up talking about broad scale pattern of diversity across entire continents. And then studying these ant communities at different spatial scale revealed that different processes were acting at these different scale, but really to understand what's happening even the finer scale, you do need to understand also what's happening at very large spatial scale, right? And so a lot of what I'm going to talk about today is about temperature, because it turns out temperature is really important for ants, and temperature mediates local coexistence in local community. It also drives regional diversity gradient along environment, elevational gradient, and temperature mixed with evolutionary stasis has an impact on the structure of ant communities across a continent. So how do ants coexist in a community, right? Why do you have more than one species of ant? Because you could think that, you know, you could have only one species of ant anywhere. Same thing with trees, same thing with amphibians, with any organism really. Why do we have more than one species in one given place? There's got to be some mechanism by which many species can co-occur and coexist. And there's many theories about why that is. And when I started my PhD many years ago, reading ant paper, many people were talking about what we call trade-off. So trade-off means you cannot be good at everything. If you're good at one thing, you're bad at another one. So this idea that coexistence in a community is maintained by the fact that you cannot be good at doing everything. And in ants, the paradigm back then was mostly about competition for resources. So it's this idea that competition is very, very important. And so you cannot be, so if you're very good at competing in one way, you're probably not very good at competing in another way. One way of thinking about ants in a more simplistic way is how do behaviorally dominant ants coexist with behaviorally subordinate ants? What do I mean by that? Well, you know, in plants, if you try to understand why some species of plant are dominant, you might not go and think so much about one tree trying to beat up another tree, right? But with ants, actually it does happen. Ants will fight. They will have territorial wars and they will destroy each other. And so and they do that for resources and for nest space. So then when you have a really big mean ant that can dominate and be very aggressive, how can other ants manage to coexist in this area, right? Because they do coexist. So that's what a subordinate ant is. A subordinate ant is an ant that is not very dominant. So there's got to be some mechanism that enable these subordinate ants to coexist with the more dominant one. So Ed O'Brien and Don Feener published this paper in 2006. That's actually the year I started my PhD, showing that in the desert of Arizona, if you look at all the ants that are there, you actually see this negative relationship between how dominant you are behaviorally, so like think of it as how mean and aggressive you are, and your discovery ability. Discovery means how good are you at finding, locating food resources and getting them back to your nest quickly, right? And so there's this negative relationship, this perfectly well done trade-off. So if you are a mean, aggressive ant, right, you're actually really bad at finding resources. It takes you a long time to find them. But then if you find them, you will displace whoever is there and monopolize the food. If you're a subordinate ant, right, the one that are at the bottom of this dominant score, then you're actually going to be often the first one to locate these food resources. So you can kind of exploit them until somebody bigger came in and kicks you out. That makes sense? It was very interesting, but you know, it was done in the desert of Arizona. As you know, deserts are not necessarily the most common or widespread ecosystems in the world. It was unclear to me whether this was representative of what ants should be in general. Is this something general about ants? It's just something that happens in the desert of Arizona. Because they were making a pretty big statement in that paper back then about this being a general thing. So I was working in the southern Appalachians in southeastern United States. My PhD was in Tennessee, as Shiva pointed out earlier. And so the Appalachian Mountains are this mountain chain that follows the eastern coast of North America. They start from, well, eastern Canada, where I'm from, actually, and they go all the way down to southeastern US. Georgia is where the Appalachian ends. It's a very old mountain chain, and so they're actually very flat and low because they've been eroded by several glaciations and other natural processes. And so since I was doing a PhD in the southern part of the Appalachians, I was like, well, I'm going to go out in the field and test this trade-off idea of LeBron and Fiener. Is it going to work out in this temperate forest? Because, I mean, temperate forests are actually a really widespread ecosystem. In North America, there are an analogous type of ecosystem a bit everywhere on the planet. So maybe if we find something there, again, that's the same trade-off, we're starting to get closer to a general trend here. So, getting some pitfall traps out, the ants that we're walking by, those are the main players that we found in that ecosystem. There was this ant called Formica subsericea, which is a competitively dominant ant, form these big colonies, thousands of workers. The workers are big, they're fast, and they're mean. And they dominate food resources. On the other hand, you have these three species that were way more subordinate in terms of behavior, meaning they would easily get displaced by Formica subsericea or other dominant ant. I'm not showing you all the species. There were about 10 to 12 species. I'm showing you the 4 ones that were the most common. And so the subordinate one include Ephinogastro rudis, Patrochinum physonensis, and a couple of species of Myrmica. By the way, as far as I can tell, all of these genera are also found in Asia, at least in parts of Japan and eastern China. I'm not sure about India, though. Do you know? Do we find all of them? This is called Nylandarian now, though, and they're not here. Anyway, so again, the idea was like, how do you coexist with this one here? And if you look at, so I basically replicated the exact same experiment that LeBron and Fiener had done many years before in the desert and found this pattern. What they found, right? Well, here it's switched, right? I put dominance on the X, RV on the Y. But in any case, what you would expect is a negative relationship if there's a trade-off. What we're finding is actually, well, this is bad science because I've put a line here, but I should not have put a line because the relationship is not even significant. There's no trend, there's no pattern, there's no relationship of any kind between dominance and discovery in this ecosystem. So no, that this dominance discovery idea cannot explain coexistence of ants in the temperate forest. What is it then? Is there any other kind of trade-off that could explain how subordinate ants coexist with dominant one? And you know, back then I started thinking about, all right, so if you're an ant in the forest, and what kind of things can limit, you know, how well you can forage for resource, all right? Well, it turns out if you walk in these forests in the summer, it's really clear that there's a different microclimate in the forest. So in Tennessee, where I was for all these years, in the summer, temperatures often reach 40 Celsius. It's really, really hot. The same kind of hot you can find here, probably hotter even in Bangalore, actually. But then if you walk in the forest, actually it's very nice and comfortable. The canopy is so thick that it creates a lot of shade. But it also suggests that light availability and even, you know, heat and energy might be limiting for things that are, you know, ectotherms that need this heat to actually forage. So I looked at the relationship, between dominance and the minimum temperature at which species were foraging out there, right? So each time I saw one of these ant species foraging outside, I had what we call an infrared temperature gun, that takes temperature measurement on the ground, tells you what is the temperature that the ant's experiencing. And after doing that for a full summer and averaging values of the minimum temperature at which these things were foraging, there's this clear, nice relationship where if you're really dominant, you forage mostly in the heat. And if you are subordinate, you forage, well, first of all, you forage at a broader range of temperature, but you can deal with much cooler temperature also, right? So, cool thing is, it does seem like there might be a trade-off here promoting coexistence. However, it was not the same trade-off as LeBrun and Feeder suggested. Though it might be related in many ways, because if you can deal with a lot of different temperature, you might be better, you may be out there foraging for a greater part of the year, a greater part of the day also, and therefore accumulate food resources for more time. And so therefore, even though you might not be able to protect these resources, at least you can get them while the dominant ant might be in their nest because it's too cold outside. In summary, it does look that in these forests and communities, there's a dominance temperature trade-off that's promoting coexistence. And so if you try to link this to climate change, it'd be interesting to think about what would happen if you warm things up, right? Well, potentially you would allow these dominant ants to spend way more time outside foraging. And so if it's the case, they could potentially have a negative impact on subordinate ants and potentially disrupt this coexistence mechanism, right? But really, before we can make such claim, we need to know more about these trade-offs. And so that's something that's lacking right now, is that we only have a few data points in the world. There's a few more done in Spain and in Europe, but we lack kind of a global, generally replicated study asking about these trade-offs in many different places in the long gradient so that we can generalize something about these coexistence mechanisms and make prediction about how climate change might affect them. So actually, you know, I've always been open for a PG student to come and work in my lab and do such a thing. So if it's something you're excited about, you know, you should shoot me an email later on. Temperature drives regional diversity gradient. So the second part of this talk is about how diversity varies along elevational gradient. As you now know, a lot of my work was in this Great Smoky Mountain National Park, and trying to understand not only if you go to low altitude sites, why there are many species of ants, but trying to understand why are these number of ants going down as you go up the mountain. That's a question that's been asked for many organisms for a long time. The cool thing about the Great Smoky Mountain National Park is that temperature and precipitation are not correlated in space. So a big problem when we try to relate climate to richness and infer mechanisms driving these patterns is that all the environmental variables are correlated. Temperature is correlated to precipitation, and these two things are correlated to productivity. And so it becomes very hard to disentangle their separate effects. But the Smokies are a great experimental playground because as you go up the mountain, temperature drops off linearly, but precipitation are actually not changing that much. And even if they are changing, there's no relationship with elevation. And there's also not a very strong variation in productivity. So working along this gradient, you know that the main climatic variable influencing your communities is temperature. And so in this paper that we published with Nate Sanders in 2007, we basically showed that temperature is the best predictor of variation in ant species richness, or we can also call it species density, along this elevational gradient. Temperature alone accounted for 50% of the variation in ant richness among communities along the gradient. So that means that if you know the temperature at a given site, you can, you have a 50% chance of predicting what's there, or the number of species that's there. Temperature is a very strong predictor. Alone can explain half the variation in richness. So that was great. But again, when you see that, you wonder, what's the other half, right? You can explain 50% with temperature, what's the other 50% that we're missing, right? And what I thought back then is that, when you work with communities, right, along a gradient, you're dealing with two different kinds of processes. Your community is in some kind of elevational band that has a given temperature, right? And will affect the number of species of ants that are there. But then where you put that community within that elevational band might also have an impact, right? So could be a southern slope or a northern facing slope, right? And you might get different number of species even though you have the same elevation. Could be in an open forest gap, or you could be in a closed canopy forest. pH might vary, whatever. Lots of things can vary with that band, and that might be the other half that we're not explaining here. So what we did is that we manipulated factors that had been shown to be important for ants in other systems. So microclimate, right? This idea that if you have an open gap in the canopy, you will have more sunlight and more heat on the ground, and that might give you more ants in that patch than in an adjacent patch where there's covered, So we wanted to manipulate this climate, and then resource availability, something has been shown with ants. As I said earlier, competition for resources, the paradigm. And so potentially by adding food resource, you could increase ant abundance and potentially ant richness. So the idea again was to try and find a way to assess whether there was some kind of interactions between regional processes such as regional climate in an elevational band and other more local processes such as microclimate and resource availability in shaping the local ant community. So how would you go about testing for this, right? I mean, of course, one way to do it would be to just go and in each time you sample an ant community in a transect, you measure the microclimate there, or you measure the amount of resource availability. But guess what? I mean, measuring resource availability in ants is not easy. You wouldn't know where to start. And I mean, it is the same for most organisms. So the best way to deal with this is to actually manipulate yourself the resource availability and ask, well, if I add resources, do I have an impact? Same thing with microclimate. It's the same problem we talked before. You can just measure the microclimate, but then which variable are you going to measure, right? And then these variables might be correlated, and so it's confounding. So why don't we just manipulate this microclimate? So I came up with this idea of having an experiment that we would replicate at many elevation along that elevational gradient. And so we had these 2 meters by 2 meters plot, and some of them were control plots, so nothing happened to them. And some of them we tried to reduce temperature, and in some of them we tried to add food. How do we reduce temperature? That was quite a big enterprise. We had these metal rebars, which are, about like this and pretty heavy, and we had to carry those all over the mountains with PVC pipes and creating these shade structures. So, I mean, actually I call that a PVC table. Then on this PVC table, we use the shade cloth. Shade cloths are usually in nursery for plants to create shade. They're great through so you don't lose your humidity. And so that's what we did, and we actually managed to reduce temperature on the ground by an average of 3 degrees Celsius along the elevational gradient. But, I was talking with some of you in the last few days about working in the Himalayas, and I mean, some of you are studying ants in the Himalayas, and I'm so impressed with you in climbing these crazy, huge mountains, and, doing field work over there in difficult conditions must be very difficult. Over there, I had access to roads, and I could go pretty close to these field sites, though I still had to do some hiking. What's been very difficult is that I tried to do an experiment. So I actually had to carry, hundreds of pipes and metal rebars in the backpack and go up and down the hill. That was the big challenge. So doing an experiment, you go another step further in terms of challenges. So I dare you to try and do that in the Himalayas, and if you do it, I want to be there to see it. So food resources addition. How we did that, we actually bought dog food because it's cheap and a lot of nutrients, fat, sugar, fibers that ants will go for. And you know that the pebbles of dog food are quite big and the ants are not really able to chew on this. So we use what's called a wood chipper. It's something you usually use to make wood chips. And we drop the food in it and grinded it to make it the finer particles. And that's what we added to our plots every 2 weeks to increase food resource availability. All right, so we had temperature reduction in some plots. We had food addition in others. And then this is metal experimental design I showed you, right? Temperature reduction, food addition, control. And we replicated this 15 sites between 400 meters in the Smokies. 400 is pretty much the lower you can go before you start getting a lot of disturbed ecosystems, right? There's lots of settlements down there. You have farms, you have roads, you have invasive plants, you have all kinds of stuff. But above 400 meters, you clear. So that's why we. And 1,300 meters is pretty much the highest peaks that you find in the Smokies. So we replicated that study, experimental study. That lasted for a full summer, so 3 months. Every 2 weeks, we'd come back and add food, measure variables, sample some ants, right? And at the end, we had some cool results. And what were our predictions? So those are not results. Those are just the predictions that we had. One thing we thought is that maybe one thing we'll find is just this one effect of shade. So if you reduce temperature at each of these plots along the elevational gradient, you will reduce the number of ants or the number of ant species all along the gradient so that you'll always have fewer ants than the control plots, right? That's one possibility. And if that's what you find, that this is arguing that, okay, you got temperature does matter at the regional scale, the band, the elevational band, but variation in micro scale temperatures also matter, right? Another possibility is that by adding food, you would increase richness. However, we thought that increasing food availability would only increase richness at low altitude, low elevation, where ants can be active all the time because it's always hot, right? Whereas we thought that at a high altitude, you can add food as much as you want. It won't really increase ant abundance or richness because temperature conditions are harsh and they will be limiting to the ants. They won't even be able to take advantage of these resources. So those are two possible predictions, right, arising from this experiment, and they would have different implications for the regional and local factors affecting in richness. So this is what we actually found. All right, so misleading again, I should probably not have put that red line because it is not significant, right? The food treatment here is non-significant. You want to look at this ANCOVA table, right? ANCOVA means that we basically look at the effect of food and shade after controlling for elevation, because we knew elevation was important. So after controlling for elevation, you do have an impact of shade. You do not have an impact of food resource, and you don't have an interactions between food and elevation either, which was my second prediction. And shade also does not interact with elevation. So the only effect that came out is shade. So it's saying that reducing temperature at a micro scale does reduce ant species richness, and it does it evenly along the gradient. Because if it was not even, you would probably have an interaction term that would be significant here. But food had no impact whatsoever. So that's the model, the prediction that was supported, is this idea that ant richness really responds to these large-scale temperature variation, but also from patch to patch variation temperature, which can be quite big, actually. As you can see, if I was able to reduce temperature by 3 degrees Celsius with the shade table, it suggests that if you're in an open gap versus, oh, that's the one thing I forgot to mention. All of these experiments were conducted in open forest gaps, so places where big trees had fallen, and so there was lots of sun availability, right? And so that's why reducing temperature made sense. And but this also means that if you were to go in the forest nearby, probably the temperature on the ground is 3 degrees Celsius lower on average. And temperature seems to be important for ants. As we can see, for coexistence, it is important. And there might be other things going on there with energy availability. All right, so both regional and more local scale variation in temperature matter. The last thing I'm going to talk about is, again, going one scale bigger, right? We started with one local patch in the Smokies. Then we went and looked at elevational gradient along the mountains. And now we're going to extend this, the grain, or now the spatial extent of our study even more and go to a full continent, right? What drives variation in ant community structure across all of North America? So, a few years ago, 2009, part of the Nate Sanders, we basically, got at a meeting all together with ant researchers from all over the world, including Europe and Australia and Asia, Africa, South America, and asked them, hey guys, we all kind of sampling ants the same way with pitfall traps and wrinkler sacks and a standardized protocol. Why about we all share our results? Why about we all share our data for the last 20 years and then try to ask some big questions with these data? And in that paper, that's what we did. So we had, several hundred sites for on ant species richness. We tried to explain what best predict ant species richness. And guess what was the best predictor again of ant species richness globally? Temperature. Right? That's exciting, but what was even more exciting is this finding that temperature was not doing as good as a job predicting richness in the northern hemisphere as it did in the southern hemisphere. Okay, that's something that ecologists have been talking about for a long time. Bob Reclef was actually one of the first ones to talk about this when he compared mangrove community assembly in North America and Southeast Asia, if I remember correctly. And Bob Reclef called those things diversity anomalies, right? He was asking, why is it that even though you have the exact same temperature in parts of Southeast US and in parts of Southeast Asia, you have way more species of mangrove trees in Asia, right? And he argued, well, it's because of different and evolutionary histories. The history of diversification extinction has been different in Southeast Asia. And so he called these things diversity anomalies, places where even though you have the same climate, you have completely different number of species for a given clade, right? So why is it for ants that you find, given a temperature, you find less ants in the northern hemisphere than in the southern hemisphere? So all of this to say, I started thinking, well, in this paper we argue it's potentially something about the history and the evolution of the clade in these regions. But why? What is it exactly? We don't know. So I started thinking about how could we go about, addressing this question about, how do you quantify the influence of evolutionary history or evolutionary history basically on things like ants? Not obvious. But back then, it was the emergence of this field of community ecology that is now known as community phylogenetics. It's this idea that by superimposing phylogenies with compositional data, you can gain more insight into how evolutionary forces have shaped the assembly or the structure of your communities. Put it simply, it's using phylogenetics to understand better what drives the composition of species in a community. It was first applied to plants by Cam Webb, Dave Ackley, Mark McPeak, and Mike Donahue. And actually, it's funny, I'm just that now I know all these guys, and I didn't know it until recently. Mike Donahue was here for the conference just a few days ago. And I also am realizing all these guys were working on plants, actually, but Mark McPeak was working on the dragonflies. I'm not sure how they got to work together in this paper together, but this paper has been cited over 1,000 times now. It's had a huge impact on our field. That's why I'm talking about it. And I think that the pioneer work by Cam Webb, trying to test some of these ideas, was working in the pterocarp forest in Borneo, if I remember correctly. Is that right, Shiva? Yeah. So not too far from here. So try and borrow some of the ideas that were in and try to see if I could use this method to understand how evolutionary history is affected ant communities and what were the important factors shaping ant communities. So the way that Cam Webb and these guys use these phylogenies is by saying, all right, we're going to take communities and we're going to map the species in phylogeny, and we're going to see, you know, in a given community, are all the species closely related, you know, or are the species actually kind of further apart in the phylogeny that you would expect just by chance, or are species just kind of a random pick of the phylogeny, right? So one of them is what we call clustering, if all species are closely related. Another one is even dispersion, if the species are distantly related. And the other option is that it's random, right? There's no phylogenetic structure in your community. Now, the way that they've interpreted this pattern, or they told us we should interpret this pattern, and this paper has been cited over 1,000, I mean, more than 1,000 times now. So people have been using this framework a lot to try to understand nature, to try to understand how communities work, right? And the interpretation that they had from it is what most people have been using since then. And since then, there have been a lot of criticism of it, and there's some flaws that have been shown to it. But I just want to start by telling you that the big main idea is that if you have this evenly dispersed pattern, meaning you pick species that are distantly related in your clade, right? If you find these guys together in a community, it suggests that during the assembly of your community, right, species that were too similar, that were too closely related, potentially that were too ecologically similar, could not coexist, right? And so you ended up with a community that looks like this. And if you do find this in nature, it's evidence that competition is the driving force of your community structure. Then they argued, however, if you do find this pattern of clade, meaning that you pick up species from the same clade, you know, or even from the same, let's say, genus within the clade, well, here you have something different happening. It's probably that competition is less important. What actually matters is that you're in some kind of harsh environment, and in order to make it in that community, you need to pass some kind of strict environmental filter, and only a certain clade has evolved the necessary adaptation to persist in these kinds of conditions, right? So this clustering would be evidence of environmental filtering, which is another process, right? So they kind of came up with this dichotomy of communities that are either shaped by competition or environmental filtering, or if you don't find any of these patterns, then it's probably just that stochastic processes, random chance is more important. Before I talk to you about how we do community phylogenetics with ants, I thought that I would talk a little bit about the actual ants again. And so these are all pictures taken from Alex Wild website. He takes really amazing ant picture. And so ant is actually one family. Probably most of you have never realized that, right? But all of ants are part of one family called Formicidae. That family has over 20 subfamilies. So those are the bigger division of the ants, the subfamilies. So these are the subfamilies that are present in North America. There's actually more than that, but those are the most abundant or common one. And quickly down of who they are. Here you have the base of the phylogenetic tree, so the more primitive ants, and you can have these very crazy, aggressive mandibles, which that they are all predators. A lot of them are actually specialized predators. So they will, things like Embryoponinae, for example, some species will only eat spider eggs, and that's it. They're specialized spider eggs, predators. They also have weird community structure. The most ants that you've heard about have this queen with all the workers, right? But actually, these primitive ants have often nest with just queens. So like 10 queens and that's it. And these queens will be seen walking around foraging, which is something that queens will never do in the most derived or more recently evolved ant lineages, right? Paraponerinae is famous. It includes a species called the bullet ant. So bullet ant is one of the biggest ants in the world. So actually in Asia, you do have bigger ants than bullet ant. But bullet ant, what they do is that they also sting. And it's been shown that the sting of bullet ant is the most painful sting of any insects in the world. It's extremely, extremely excruciating. So these are my bullet ants. Icitoninae are the army ants. These are ants that form nomadic colonies. So instead of putting all their ants in one nest, they actually are on the move all the time. So you have millions of workers moving all the time. And they are very, very voracious. They will eat anything that is along their way. So if there's a frog, a bird nest, a snake, a small mammal, they will eat it up within minutes. Very aggressive. Pseudomyrmicinae are more famous for being associated with acacia trees. And then finally, you have these guys, dolichoderines, pharmacines, and myrmecines, which are very successful ant subfamilies, especially pharmacines and myrmecines. Again, I was looking at the ant collections that you have here today, and the ant collection was dominated mostly by these two subfamilies, with a lot of those also. But definitely North America, most ant communities are dominated by these three subfamilies. Okay? So that's just a little bit of ant natural history. So that's the ant phylogeny that I was able to put my hands on back then. It was phylogeny was already published by Sean Brady et al. It was a genus level phylogeny that I've modified to make it a species level phylogeny using introducing some terminal polytomies in it, and so that I ended up with about 300 species in it. And again, it shows you that these are the basal lineages that includes those specialized predators. And then at the tip of the phylogeny, you have pharmacines and myrmecines, which are more recent, and are more generalist, has omnivorous diet, and often form association with plants or with insects feeding on plants. So that's just summing up with plants. So I had this access to this phylogenetic tree. It was integrated phylogeny, and therefore I had branch length, and I was able to use it to look at the community structure, phylogenetic community structure of ants. So and then I spent about 2 years getting data from the literature on all the studies that had been done on ants in North America, and then extracting list of species for each of these communities, right? I had about 300 of them. So then I was able to say, using the phylogenies, I was able to quantify the phylogenetic structure of ant communities in each of these yellow dots. How do you calculate phylogenetic community structure? You can calculate this thing called MPD, mean phylogenetic distance, which take all the species pairs in your community and then calculate the distance in them. So in the phylogenetic tree, you know, how many million years of divergence separates them. Then, so you take all the species pair in your community and you average those distance for all of them. So you get an average distance. If the distance is big, it suggests that there's even dispersion. If the distance is small, it suggests that there's clustering. Remember? So that's what you want to know. But again, the thing is, you cannot just describe this pattern. You need a statistical test. You need to ask, all right, so this looks clustered to me, but how do I know that it's significantly clustered? How clustered is clustered, right? You need to answer that question. So the statistical test you can do for that is called a null model, all right? A null model is this idea that you will create random communities. So you'll take, you create random communities, pick ants at random, and then calculate MPD from these ones, right? And you can do that 1,000 times, so you get a distribution of null community values, so random MPD values. Then you can compare your observed MPD for one of these yellow dots to the distribution of these null values. And hopefully your value will fall outside the null distribution. So if it falls outside the null distribution, it suggests that your community has a structure that is different from the null, right? That's your test. And so then you can say, yes, it's actually structured. Not only you can do that, but you can calculate the strength of that deviation, right? So these, on the x-axis here, you have the MPD value. You can think of those as million years of divergence on average between species in your community. This is a hypothetical example, my observed value fell here at about 37, right? Then I created null values by saying, okay, I have 4 species in my observed communities. I'm going to sample 4 species at random from my entire phylogeny, right? And get an MPD, a new null MPD value. I'll do that 1,000 times, and I end up with the distribution of null MPD value. So you can see that the mean of around 29. So yes, in this case, you're way outside the null expectation. You have greater phylogenetic distance on average than expected by chance. So here you could conclude you have significant even dispersion. You could potentially conclude there's competition shaping that community. But really what I'm interested in here is that I have these 300 communities. So I don't want to just say, yes, this one is significant. No, this one is not. Yes, this one is. I want to synthesize all of that. So what I did is I was interested in the standardized effect size, meaning the deviation. How far is this line from the mean here, right? Then the idea is to try and relate this to variation in environmental variable to say, why? Are there some places where competition is more important? Are there some places where environmental filtering is more important? Just to refresh you, evenly dispersed looks like this, clustered looks like that, right? So if you're evenly dispersed, you're on this side of the distribution. If you're clustered, you're on this side of the distribution. Then the metric that, there's a metric that you use to, the standardized effect size metric is called NRI, Net Relatedness Index. And so here, instead of putting the net relatedness index for each community, I color-coded them so that the blue colors show you communities that have clustering. So they have large value of net relatedness index, and even dispersion shows you, the red values, orange value shows you even dispersion. As you can see, so in the southeastern part of the US, which is pretty much tropical in some ways, it's kind of the convergence of tropical Florida and temperate North America, right? So you have some tropical lineages down there. As a matter of fact, you find even dispersion, whereas as you go up north, you tend to find more clustering. So you have this general pattern of, as you go from warm to cold and wet to dry, you go from evenly dispersed to clustered. This is a different way of looking at it, just a bit more stimulating way, I guess, to look at it. But if you were to summarize all of this data, at the bottom left here, where you have cold and dry temperature, you have clustering. When you have more warm and wet temperature climate, you get even dispersion. So you get this gradient, right, of phylogenetic clustering along a kind of tropical to temperate climate gradient. So what does it mean, right? So if you go back to Cam Webb et al. Ways of explaining this pattern, it would basically, the suggestion would be that, okay, well, in the tropics, competition is more important in the assembly of community. But then as you go up north in Canada, where you get freezing in the winter, drier condition, well, up there, environmental filtering is actually what matters. That's one way of thinking about it. All right. Turns out, though, that in this paper, non-model analysis to try and see if this pattern holds after you account for these regional processes. Remember, I talked about biodiversity anomalies earlier. So, so Rick Clef was saying that there might be different number of species in Southeast Asia than in North America because evolutionary history is different, and evolutionary history is shaping the species pool, right? So I've done some analysis that I'm not presenting here because it's a bit too complicated, to basically say, if I account for regional processes here, and if I account for regional processes there, do you still get these patterns? And you don't, okay? And so what this means is that, so basically this pattern became all yellow. After I accounted for regional processes, all of this became yellow. So that suggested that all of a sudden I captured the process that was really important. And that process was not a process that operates at the community level. It was not competition preventing species locally from coring to one another. It's something that happens at broader spatial scale. So what might that be? What is it that might have shaped the regional species pool, really? If you look at who's there, right, what you find is that in more tropical site, you draw species from all over the clades, all over the phylogeny. When you go up north, you get only Formicines and Myrmecines, the two most derived groups in the phylogeny, right? These guys at the bottom. Well, this is very much aligned with a theory that, well, a hypothesis actually that came out that was suggested by Weins and Donahue in 2004 in a Trends in Ecology and Evolution paper, in which they in which they describe a pattern. I mean, this is just one of the examples they gave, but they argued that this pattern is found across broad taxonomic group all over the world, and is the pattern of having ancestral clades trapped in the tropics and more derived clades concentrated or being able to colonize northern regions. So what might be the explanation for this pattern is this idea that most lineages that we know of today have evolved in a largely tropical world, right? But in around 30 to 40 million years ago, there was a major cooling of our planet, and then we started getting more temperate type of ecosystem, temperate forests, boreal forests, grasslands, prairies, and such a thing. And so there's been this major switch in climate creating all these new ecosystems, right? And they're claiming that only a subset of species, only a subset of lineages have been able to actually jump in there and disperse and diversify in these types of ecosystem. And they've called this the tropical niche conservatism hypothesis. Is this idea that because most lineages were originally tropical, most extant species have retained these tropical adaptation and struggle with anything that diverge from a tropical condition. So when it gets dry and cold, most things struggle, especially if you're an ectotherm, right? And so that's the same pattern we're finding with ants, where the more basal groups are stuck in the tropics and the more derived ones have managed to disperse and diversify in northern regions. So it seems that what's happening here is that really ant community structure is the result of having different species pool. One that is have tropical clades plus recent clades, and another more northern species pool that has only the most cold adapted clade. And it's the signature of these different species pool that you're seeing in the community level. It's not something that's happening in the community. It's something that's happening. It's something that's happened over millions of years that's shaped your regional pool, and the signature of that pool is reflected in your local community, right? So it's like saying tropical niche conservatism trickled down to affect the structure of your local community. Yet, if you think about it, once again, temperature does matter, right? Because temperature is probably one of the main selective forces here shaping this pattern. So, in summary, conservatism, evolutionary conservatism, impose a constraint on adaptation to and diversification into novel environmental niches, especially the cold niches. So then changing contemporary climate can also interact with this conservatism, right? Because ants might or might not be able to adapt to this heat we're creating, depending on whether they're cold adapted or not. So that's a summary of my talk, is this idea that to understand the local community of ants, probably of everything, you really need to study these organisms to know, go in the field, do detailed studies locally, understand the behavior, coexistence mechanism at very fine spatial scale. But that is not enough to make some broad generalization. You also need to replicate your studies along broad spatial scale and study communities along broad scale gradient to see how community dynamics interact with broad scale abiotic gradients, but also with long time evolutionary processes. So it's this idea that you can see communities as an onion with different layers, and you can add the global, continental, regional, and local filters or processes, and these give you the structure of the community you see today. but you need to really need to have this outlook that goes across spatial and temporal scales. So I won't repeat this again because I said it many times, but you know, I guess the conclusion here is that temperature seems to matter for everything in ants, and I'm sure it's the same in other ectotherms. And so we can only imagine how warming up our planet can affect these processes, which in the end are generating and maintaining biodiversity through time, right? This biodiversity we're trying to conserve. So yeah, some thinking, some food for thought here. So I have to thank all of my collaborators and all the undergraduate students that volunteer on the project and the several funding sources. Thank you so much for your time. I know it was a long talk.